Today I'm going to talk to you about my research that I've been doing all summer. What we're primarily looking at are the current status of freshwater mussels in Cedar Creek, and we're also looking to gain a better understanding of their relationship with macroinvertebrates. Now, that relationship might not seem very exciting, but because we don't know very much about unionids, we don't know about their life history, not all that much about their life cycle or their habitat needs, being able to have a link between something we know really well and something like unionids that we don't know all that much about, that only makes us gain a better understanding. We can use that understanding to apply different conservation methods to help them survive during this hard time for them. Now, there's different anthropogenic influences that's caused the drastic decrease in the increasing endangered species list to grow so much, especially lately. It kind of all started with the button industry. From the 1800s to about 1940, people primarily used freshwater mussel shells for their buttons because they're pretty, they're strong, they're durable, and they're very easy to collect. There was a huge spike in the early 1900s on freshwater mussel farmers and people who would go out and harvest these for the different button industries. Also, poor water quality due to runoff, or especially storm runoff, kind of affects the stream in a whole, more so than mussels. Stormwater runoff is when it rains a lot, all this water, because there's so many hard surfaces like the streets, sidewalks, buildings, covering all of the natural soil and plant roots and all of that, those aren't able to take in the water. So the water runs off of these impermeable surfaces a lot faster, causing more contaminants to get introduced into the water system. Also, dam construction and destruction is more of like a modern concern versus a historical concern. A lot of dams were built, but taking out a dam will wipe out a whole population just because excuse me. It changes the whole benthic layer, all of the different substrate compositions in the stream, and because mussels are sessile, they cannot move to a better location. Different conservation methods that are being looked into are relocating these freshwater mussels, especially the endangered ones. The only problem with that is a lot of people don't realize that they need specific life cycle requirements, like host fish. They need certain habitat needs, like different composition of substrate that they live in, and also invasive species. Invasive species were introduced to the Great Lakes about mid-1980s. They include the zebra mussel and the pilot mussel. Now, these are a lot different from unionids. They're smaller. They don't get any bigger than about 2.5 inches long, and they do not need, they don't really have habitat requirements because they can latch onto anything, and they're very tolerant to pollution. So because of this, they can take over biomass of almost just about any kind of water body, which includes streams, lakes, rivers. They latch on to factory pipes, all of that, so they cause a lot of money and damage, and they also cost a lot of money to remove them. They're really hard to get rid of, and as of now, their conservation is not their conservation. Everyone's trying to get rid of them, but because they can lay up to 1 million eggs per spawning season per individual, it's nearly impossible. So what we're trying to do is conserve all of the other water bodies that have not been introduced to these invasive species quite yet. The unionids are a lot different than the invasive mussel species. They are pretty intolerant to pollution, so if there's a pollutant introduced, the population wipes out because they cannot survive in that kind of hostile environment. Now, unionids also have an interesting life history cycle. They require what's called a host fish to take care of their parasitic larvae, or their Wachydia. What's shown in this video right here is a lure, a freshwater mussel lure that attracts host fish. Not every mussel species has this lure. Some just disperse their Wachydia into the water column, hoping they will find a host fish to latch on. This lure in particular is really interesting. I really like looking for these. But this lure mimics their host fish's prey. So it acts like the host fish's food. The host fish will latch onto it or bite it, thinking it's food, and the mussel senses it and ejects their glaucidia into the fish's mouth and surrounding gills and fins, where the glaucidia will latch on until they are mature enough to filter themselves on their own. Macroinvertebrates are very well known within the aquatic ecology community. They are great bioindicators, which means they can biologically indicate whether or not a pollutant has been introduced just by knowing if they're present or not. So what's shown on the right is a water penny, and the bottom right is what's called a damselfly nymph. They're very intolerant to pollution, so if they're present, then you're good to go. Crayfish are very intolerant. They're very tolerant to pollution, so it's pretty common to see a crayfish, especially since they're predators. They don't necessarily need good water to eat and feed themselves. Now, our study site consists of Hastings, Michigan. It's about an hour and a half south from Grand Rapids. We're doing our study in Cedar Creek because there was a previous study done 3 years ago by Dr. Dalen Woolnough in Central Michigan University, looking at their current status. We wanted to choose this site because we know that they are present and they had a rich diversity 3 years ago. So part of the work on conserving an endangered animal in general is checking on the status and checking on their population. We did 2 reaches, which means we did 2 long segments of the river, 100 meters each. We did a total of 18 samples, randomly chosen, with a randomly chosen chart. And yeah, 36 square meters of mussels were surveyed and calculated, not per site, but total, and about 20 square meters of macroinvertebrates were sampled. Our methods included for macroinvertebrates using what's called a HESNet sampler, which is shown right here in the picture on the left. What it is a bottomless bucket that has a screen to let fresh water in the current to continuously flow through the bucket without letting in new particulates. And then we have a triangular net that gathers to the bottom where this container is. How to use this particular net is to mix up the substrate, scrape off the insects off of the rocks and the shells, everything that's in that bucket. And it will all drift in this container where I'll add it to a container with ethanol and I'll analyze it in the lab. Now, because unionids are endangered, at least a lot of the species are, especially in this creek, the Department of Natural Resources required us to visually inspect and identify and replace them back in the field. So unfortunately I don't have live specimens for you guys to look at, but I do have shells, which I think you guys might have looked at earlier. Visual inspection included, depending on the density, this particular site had a very high density of live unionids. So what we did is we took them out, put them in the creek bed, and identified them to species. After a while, after I got the hang of identifying them, because they were pretty difficult to identify, what I did was I snorkeled and, without completely taking them out of their buried state, identified them like that. That way it went by quicker and I didn't disturb them nearly as much. Now, aquatic macroinvertebrates can be grouped into 4 basic functional feeding groups, which is characterized according to how and what they eat. So it really depends on their behavior. So shredders, gatherers, scrapers, and predators are the 4 basic functional feeding groups. Shredders eat what's called coarse particulate organic matter, which includes leaf litter and large woody debris. Gatherers, including filter feeders and gathering collectors, eat what's called fine particulate organic matter, which can include fecal matter, it can include leaf litter, nutrients in the water column, that sort of thing. Scrapers eat algae off of the shells, off of rocks, off the wood, off of pretty much any surface, and then predators obviously eat prey. Now you can manipulate these 4 functional feeding groups to do an analysis of a general stream quality health. So for example, you can make scrapers to shredders plus total collectors, and you can get analysis based if the stream is heterotrophic or autotrophic. For the data I've collected, we found that the stream is mainly heterotrophic, which means it obtains its food and energy by taking organic substances, such as plant or animal matter. We also found that channel stability is very high based on the number of filter feeders present. So they plant themselves on rocks, on the shells, anything hard that they can plant themselves on. So there's a large number of that kind of tells us the substrate channel stability. Also, for top-down predator control, we found that there's a very low predator-prey ratio. This could be because of the huge increase in filter feeders, but we just thought that was interesting. I don't know why it's like that, but it's one of my further research studies. We also did what's called a linear regression model for the population densities per site. So each individual dot is a site with unionids as the x and macroinvertebrates as the y, and we found that this data is very significant with a p-value of 0.003. Basically what this is showing is the increase, as unionids increase in population, so do macroinvertebrates, just looking at the live specimens. I thought that was really cool because that pretty much answers one of our hypotheses. Now in conclusion, conservation of any endangered species, especially unionids, is essential. If you don't do any kind of conservation work, you don't check up on the population or see any kind of environmental changes, then they have the possibility of becoming extinct. Now checking on the current unionid populations is very important as well, just like I said before. This kind of lets you evaluate land use, so you can document any recent land changes, such as if there's a farm nearby and they use excess fertilizer, it runs off into the stream and there's all this fecal matter or excess nutrients that the unionids can't support or can't live with, then that is something that we can change through regulation. Cedar Creek does not have invasive species, mussel species at this point, so further evaluating and checking up on these different streams for invasive species is also essential. Now, seeking different biological relationships is also important, especially if you don't know much about one or the other. So the relationship between unionids and macroinvertebrates can tell you a lot. Because we know so much about macroinvertebrates and preferences as far as being in a riffle or a depositional area, this can tell you what habitats that some unionids prefer over another. Also, different statistical modeling, like you've seen before, kind of helps bridge the links between